So transdermal administration, and I'm going to talk about the pluronic lecithin gel products that are not approved, is an example of a compounded preparation. And it's a very tempting preparation to use because theoretically you can apply it on a cat's ear and therefore avoid oral administration, which can be a trial. And there's other nice things about transdermal administration. You bypass oral absorption and therefore drugs don't undergo first-pass metabolism, those sort of things. The problem is the science supporting the movement of drug that's been topically applied inside the ear into the subcu where the absorption is going to occur is very limited. The skin is a jealous organ. It does not like toxins penetrating it. The skin is always constantly being exposed to toxins in the environment, and so it's got a great system for preventing compounds from getting across the strand corneum. True, it is thinnest in the inner pinna in the cat of the ear, but to overcome those barriers is very difficult. And while the lecithin organic component of the transdermal gels is designed to do that, the studies that have been done thus far suggest that the drug is not going to move across, certainly not following single dosing. But even in our studies and others, using transdermal gels at 2 to 3 weeks, when you compare drug concentrations in the plasma when administered as a transdermal gel to the same drug concentrations when you give the drug, say, orally, very rarely does it work. And we have demonstrated that it does work for methimazole. The nice thing about methimazole is you're going to monitor response, you're going to monitor thyroid response in those patients. So if there's any question, you'll pick up on it. But we've demonstrated it doesn't work for a variety of other drugs. And when it does work, it's in a small proportion of cats, and you can't really predict which cats it's going to work. So the problem with transdermal gels is, while it's tempting to say when there's no other alternative, I think that that gives us a confidence in something that really doesn't exist. And so my suggestion at this point is to limit it to methimazole or situations where you can actually monitor. You have a good outcome assessment, whether it's plasma drug concentrations or some kind of good clinical parameter that you can monitor to assure that your patient has responded.